Have you ever wondered how the genetic information stored in DNA is expressed? Is there any process for understanding the information stored in DNA? The central dogma explains this process to be happening in 3 steps, namely replication, transcription and translation processes. Replication is a process by which DNA gets duplicated to produce its copies. Transcription is the process by which the information contained in the DNA is transferred to a newly assembled piece of mRNA. And translation is the process by which the RNA codes for a specific proteins. In this video, we will be learning about transcription. To be simple, it is the process of copying genetic information from one strand of the DNA into RNA is termed as transcription. So, where does this process takes place? For eukaryotes, transcription takes place in the nucleus, while for prokaryotes, transcription takes place in cytoplasm. Transcription also follows the principles of complementarity, but it should be noted that thymine is replaced by uracil. It is seen only in the process of transcription. Unlike replication, in transcription, only one strand of the segment of the DNA gets transcribed into RNA. Why only one strand gets transcribed? Let us find the reasons. If both strands act as a template, they would produce 2 RNA with different sequences, thus producing 2 different proteins, thereby complicating the mechanism. The 2 RNA produced would be complementary to each other, forming a double-stranded RNA. So, what is the issue here? It prevents the translation process and would complicate the genetic information transfer machinery. Then, how does this complication affect the process? It prevents translation into proteins. So, for the process to be done, we have a transcription unit, which consists of a promoter region or a promoter sequence, a structural gene, and a terminator. It has got a template strand with polarity 3' to 5', and it is a non-coding strand. The transcription process is catalyzed by the enzyme, which catalyzes transcription, and the enzyme is DNA-dependent RNA polymerase enzyme. So first, let us check more on structural gene. The structural gene is a part of template strand which has a polarity 3' to 5'. It is the DNA part which forms the mRNA. The structural gene is flanked on either sides by a promoter and a terminator site. The promoter is a DNA sequence which is present at the 5' and it serves as a binding site for the RNA polymerase, while the terminated region is located in the 3', that is downstream, and it serves as a site for terminating the transcription process. So, can you name the basic functional unit of inheritance? Yes, it is the gene. My next question is, how will you define a gene? The DNA sequence that codes for an RNA molecule is termed as a gene. So, is cistron and genes are same? Let us find out. A cistron is a DNA segment that codes for a polypeptide. The DNA with one cistron is termed as monocistronic, and it is seen in eukaryotes. Whereas the DNA segment with more than one cistron is called as polycistron, and it is seen in all prokaryotes. You have to note that the eukaryotic monocistronic structural gene has both introns and exons. Exons are the coding parts or the sequences which appear in processed or matured RNA, whereas introns are the sequences that do not appear in mature RNA. So we can simply term them as the intervening sequences. Let us now check the types of RNAs. There are 3 types of RNA. First is the messenger RNA or mRNA that provides the transcript copy of a gene that codes for a particular polypeptide. They work as a template strand. Next type of RNA is transfer RNA or tRNA. They carries the amino acids to the ribosome. Next is the rRNA or ribosomal RNA. It is a component of the ribosome and performs the catalytic and structural function. The fact is that all three RNA are required for protein synthesis. Let us now check the process of transcription in both prokaryotes and eukaryotes. The process of transcription takes place in 3 steps in both prokaryotes and eukaryotes. In the case of eukaryotes, there are different RNA polymerase enzymes and the RNA processing is done in the nucleus of the cell. Now, let's check the transcription in prokaryotes in detail. The first step is the initiation process, followed by elongation and finally the termination process. So, can you recall the name of the enzyme which helps in transcription? Yes, it is a DNA-dependent RNA polymerase enzyme. So, the enzyme RNA polymerase binds to the promoter region, initiating transcription. That is, it is in the 5' to 3' direction. You know, a special structure called the initiation factor, we call them as sigma factor. It gets bound to the RNA polymerase enzyme which helps it to recognize the promoter site of the DNA which is to be transcribed. So, what is the function? It helps to recognize the promoter site for transcription. Once the RNA polymerase enzyme has reached the promoter site, the sigma factor drops out from the enzyme. Upon binding, what happens to the DNA? The DNA double helix unwinds and separates. The enzyme uses nucleoside triphosphates as the substrate and polymerizes the template strand following the rule of complementarity. RNA polymerases move along the strand and only a small stretch of RNA binds to the RNA polymerase. Once the enzyme reaches the terminator site, the polymerase enzyme gets bound to the termination factor, which is the Rho factor. You can see a Rho factor that is green in color. It helps to terminate the transcription process. With the RNA polymerase, the emerging RNA falls off and it results in the transcription termination. The DNA helix zips back together and mRNA moves to the cytoplasm for protein synthesis. So, how does the transcription and translation occur simultaneously in prokaryotes? The reason is that the mRNA does not require further processing and also both occur in the same compartment. Let us now check the differences in transcription seen in both prokaryotes and eukaryotes, right? Since the prokaryotes lack a nucleus, the process of transcription occurs simultaneously with the translation in the cytoplasm, whereas transcription in eukaryotes occurs within the nucleus and translation occurs in the cytoplasm. Prokaryotes use only one RNA polymerase for transcription of all types of RNA, whereas eukaryotes use different types of RNA polymerase, that is RNA polymerase 1, 2 and 3. Prokaryotic transcription does not involve mRNA splicing. The eukaryotic RNA transcript, that is the primary transcript, contain introns, that is non-coding sequences, and exons, which are termed as coding sequences, and is considered as pre-mRNA. It needs to be processed into mRNA containing only exons. You need to note that there are 2 additional complexities in transcription in eukaryotes. First is the different types of RNA polymerases. There are 3 RNA polymerases, each having a specific function. RNA polymerase 1 transcribes rRNAs, that is 28S, 18S and 5.8S in the nucleolus, which is assembled to form ribosomes. RNA polymerase 2 transcribes the precursors of mRNA named as heterogeneous nuclear RNA, that is hnRNA. All protein coding pre-mRNA is in the nucleus. So, the pre-mRNA as mentioned contain introns and exons, and thus it must be processed to mRNA, which can then undergo translation process. Next RNA polymerase is RNA polymerase 3. It transcribes a number of RNA such as pre-tRNA, that is transfer RNA, small regulatory RNAs, that is 5 srRNA, small regulatory, that is SR, and small nuclear RNAs, that is snRNA. There are a few additional steps to be noted. The primary transcript, that is a pre-mRNA, contains exons and introns, which are the coding and non-coding sequences, making it non-functional. Thus, the pre-mRNA must be processed to form a mature mRNA for translation. Which tool has been used to remove the non-coding regions? The transcript is spliced by spliceosomes. So what is the function of a spliceosome? It is to remove the introns and the exons and are joined in a defined order. The spliceosome is a large RNA protein complex which catalyzes the removal of introns from pre-mRNA. Further on, the hnRNA undergoes capping and tailing to protect the mRNA. The mRNA is capped by modified guanyl, that is methyl guanosine triphosphate on the 5' end, whereas the adenylate residues are added to the tail, that is a 3' end. The fully processed hnRNA is now called mRNA and it is transported out of the nucleus into the cytoplasm for translation process. Now, let us focus on the significance of the complexities. The split gene arrangement probably represent the ancient feature of the genome. The presence of introns is a reminder of antiquity. Splicing represents the dominance of the RNA world. Isn't it wonderful to know that these mechanisms takes place in each of our cells independently along with other processes? Do you want to find out what happens after transcription? Watch on to learn about the third step of protein synthesis. Now, let us summarize what we have learned in this video.
Manorama Horizon. Learning made simple.